As you can see, Hillel already put his slides on. Seems like it's going to be very interesting. Let me just introduce our speaker. So Hillel Kugler is a faculty member of the Faculty of Engineering in Bar-Ilan University in Israel since 2015. His research interests are in modeling and analyzing complex systems using formal reasoning and synthesis methods, with a special focus on the development of new computational methods and tools towards enabling a deeper understanding of biological computation and biological devices. Before joining Bar-Ilan University, Hillel was a researcher at Microsoft Research in Cambridge. And before that, he was a postdoc at the biology department of NYU and a member of the analysis of computer system groups in the Courant Institute, also at NYU. He did his PhD at the Weizmann Institute of Science in Israel under the supervision of David Arel. And Hillel, the stage is yours.
Great, thank you very much, and thanks for the invitation. And I'm happy today to have an opportunity to tell you a bit about approaches for formal verification in biology. And so I'd like to give you, in addition to a flavor of the verification, what are the main kind of questions, some of the achievements in the field so far, and some directions where I believe research might be going. So first of all, for verification to actually be applicable and useful in biology, one thing that we already take for granted, for example, in software and hardware, that we need accurate computational models. So what I mean by this, that the model doesn't have to be accurate in the sense that it captures every detail. Actually, usually it can do it, but it has to be meaningful so we can deduce insights and new facts. So models are useful if you can get some mechanistic understanding of the system, and actually building the models and figure out what are the questions is still one of the main research challenges. And as I'll tell you, in the past few years, there's been progress in terms of building models, which opens opportunities to doing the analysis and verification. But it's not the situation that we just already have a well-defined, precise models like we had in the standard applications that you're familiar, and we just need to develop the algorithm. The big question is, how to come up with a model. So a small disclaimer, in this tutorial, there's a lot of really important and interesting work that I won't have time to cover. And for people that are interested, I would recommend to look at proceedings of the annual CMSB conference, the Conference on Computational Methods in Systems Biology. This is a Springer LSCS proceedings. This is more for the biology, for the natural biology, and for the engineered biological applications, there's a conference on DNA computing and molecular program, which has a very interesting and good papers. And I think in both of these conferences, although there's like biological background, the papers are quite mathematical with definitions and proofs. So I would say it's really accessible if people are interested to learn more. And actually, I'll be happy if part of the results of this tutorial, people will find it worthwhile to go and dig deeper. Okay, so I decided to divide the applications in biology to two parts: the natural biological systems and engineered. In natural biological systems, we have we build models of either a cell or an organism, and we, like biologists, want to figure out what's going on to predict the dynamics. Whereas in engineered applications, a part of the goal is to create either new biological devices that can perform computing and then maybe either replace or for certain applications use the biology for our benefit to do computing, or in this way interface with biological material and be able to interact with biology. So this is the engineer. In this tutorial, there are many aspects of biology that can be modeled. And I'll tell you mostly today about what we call gene regulatory networks. So gene regulatory networks are circuits in which genes control the behavior of each other, and in this way, they determine the decision-making of a cell. We'll see more about it later. And I'll tell you about a tool which is called RAIN that we've been developing for quite a few years at Microsoft Research that allows to automatically construct these GRNs from data. On the engineering side, there are also many different approaches to build devices, and I'll try to briefly tell you about two approaches. One is called DSD, DNA strand displacement, and the other is an approach called network-based biocomputation. So we'll get to this later. And in terms of underlying mathematical formalisms, for the natural systems, we'll mainly talk about logical models, and which have this kind of discrete abstraction, and we'll also focus in particular about Boolean networks, where each component is either on or off, and these models are, of course, a big simplification, but they're quite powerful and also amenable to formal verification. In engineered application, I'll mention the model of a chemical reaction network, which is elegant mathematically and also useful, so we'll get to that. And also, you can feel free to stop me and ask questions also during the tutorial. So let's start with natural biological systems. In biology, the basic unit is the cell. So there are some organisms like the simple bacteria that are just composed of one cell, and it has to function on its own. And later, multicellular organisms is developed, and then within an organism, many cells develop and coordinate their behavior and need to function together. And one of the deep biological questions that people want to understand is this question of genotype to phenotype. So what does it mean? The genotype is the activation of genes in a specific cell or organism. And some of these genes might be mutated or not functioning, and we want to, or you have different variations of the genes, and we want to understand what is the effect of this genetic code on the phenotype, which is the observable behavior. So of course, in humans, it's a very complicated question. And genes affect many things, and there are many side effects, but in simpler organisms, it's easier to study what are the effects of genes. These are some of the high-level questions, and as a case study for some of the modeling and the verification, I'll tell you about a classical systems in biology, which is called VPC, valvular precursor cells, in C. elegans. So this is a classical system in biology because it kind of studies an important feature of cells, that is how cells make a decision how to differentiate into a specific cell. So there's this phenomena that initially cells in the body are quite generic. They don't have a specific function, and later during development they can become more specified and have a specific role. So we'll see in C. elegans how a family of several cells make this decision, and I think this is also, while I prepared this talk, it's quite interesting because there's been quite a lot of work over the past 15 or so years of building many different models to study the system. So it's quite instructive to look at the computational efforts. So what is C. elegans? C. elegans is a small worm that is widely used by experimentalists, by biologists. And some interesting properties that it has a fixed development, and it has these almost 1,000 cells, 959 cells, that each time the worm develops, it exactly generates this a fixed number of cells by a deterministic program, which is quite an amazing property. So already you can see that this is like a very precise machines that know exactly what to do and how to prepare the cells in the right places to the right fate. The life cycle is quite short. which means for experiments it's an advantage because you can, sometimes you want to do experiments on the progeny. So within a few days, you can get babies and then babies of the babies and check out all kinds of genetic effects. Another fun fact is that you can freeze a worm and then later take it out of the freezer and get it back to life and to do experiments on it. So this is very useful. The experiment is they can store a specific a strain with a certain mutation, and if they want to get back to it, they just get it out of the freezer or ask from some other lab that has this strain and they can do experiments. So this is a bit about C. elegans. And C. elegans and other such systems are called model organisms. And the idea is that like we usually treat model, it's a model in the sense that some of the properties that hold in these simpler systems also hold in more complex organisms, including humans. So many of the actual discoveries that were discovered in C. elegans are relevant for us. For example, programmed cell death. So this is one big discovery that initially people thought that when cells die, they just either lived a lot or because of wear and tear, the cell suddenly collapses and dies. But it was figured out that this is a well-regulated biological process where a cell gets specific signals and can be, there's a decision to kill the cell and then it goes through a process of dying. So it's not just a spontaneous death. It's a well-regulated process, and this also has implications for human biology and for cancer. In some cases, the cells they don't know that they need to die, so they continue to divide. So this is an example of one discovery made in C. elegans. Another is RNAi, RNA interference. It's a technique in which you can silence a gene. And when the gene is silenced, it means that it doesn't produce a specific protein that is in charge of producing, and this will affect the dynamics of the system. And this is very important, as we will see, for when we want to uncover these circuits, these gene networks. One of the main techniques in genetics is to try to break the system. So we silence a specific gene or another gene, and then we observe the system. We see what happens, and for that we can make inferences. Right, so it's like breaking things and figuring out what goes wrong, and from that trying to eventually infer how the circuit is connected. And the third discovery is a green, GFP is green fluorescence protein. So this is like a glowing marker that you can put within a cell, and you can connect it to a specific gene being active. So this allows you to observe your gene of interest and to make the cell glow green if the gene is activated. So again, this allows a powerful experimental methods to do measurements. Okay, and maybe the last actually fascinating discovery I wanted to mention is this genetic regulation of aging. So what you see in this graph, on the x-axis, we see this is a time in days. And the y-axis shows how many worms are still alive. So if you look at the black line, which is marked N2, N2 is the kind of the standard worm that everyone does the experiments on. It's called the Y-type. So you can see that by around a month, 30 days, all the worms are dead, right, from natural causes. Whereas if you look at the red line, which is called the DAF2, DAF2 is a mutation in one of the genes. So you can see that you have worms living up to more than 70 days. So it's actually a doubling of the lifespan of the worm by making a change to one specific genes. And you can also see in the green line, it's a combination of DAF2 plus two other genes that together have this effect of increasing the lifespan. So, so this is another interesting fact about the aging. And of course, after this discovery, now there's quite a lot of interest to see how this kind of process generalizes to more complicated organisms and perhaps to humans. Of course, it's probably much more complicated, but there's a lot of research and understanding what goes on here. And actually, this is a field where computational models still haven't made a big impact. So this is an interesting future research direction. I think that the reason where we don't have good computational models is this is a really very complex phenomena involving many different factors, and it might take us much more time until we'll be able to build these models that will help us predict a lifespan in aging. So we'll talk about simpler models of these cells, which are called VPCs, as you can see marked in red. So here in the picture, this is the lineage. This is the divisions of the cell in C. elegans. And as I told you, you have a fixed number of a bit less than 1,000 cells. And you can see here the pattern of divisions. And in red are these 6 cells that are called VPCs, vulvar precursor cells, that we're going to study their behavior. So what we see here, this is a picture from one of the classical papers in the field on this system. So in the 3 rectangles, what you see is these are 3 VPCs out of the 6 ones. And what happens is that they acquire fates. So the one on the left is going to become primary, designated by this number 1. Next to it, there will be a cell that is going to become secondary. And next to it, a cell which is tertiary. In addition, you see in the diagram the different interactions between the cells, and there's another cell that sends a signal to the other cell. So I think this is interesting. This is how, until recently, biology described their models. So they would draw sketches like that in more complicated diagrams, and then they would reason in their head about the different interactions. And many of them can do really amazing jobs in terms of remembering much more complicated pictures. But as we know, there's a limit to what we can do in our head, and it helps to have computational models. So things that are challenging are to take into account the progress of time, the concurrency between the cells, the fact that cells make kind of decisions independently. So it's a distributed control, and of course, the interaction with the different components. And as the system grows, of course, it's much more complicated. So a bit more about the VPC system. So we have in the top of the diagram, we see 6 cells, P3P up to P8P. And initially, they're all gray, which means they're still undifferentiated. They're part of this equivalence group. So in the beginning, each one of them has the potential to be either primary or secondary or tertiary, but they will make these decisions according to signals that they get. So the cell in the middle, the blue cell, is going to get a strong signal from this green cell, which is the anchor cell. It will turn a primary, which is designated by blue here. It will also send signals to its 2 neighbors to tell them to become secondary, and the cells further away are going to become tertiary. And this fates designated later how many divisions the cell will undergo and the axis of the division. Okay, so we said this is the signal from the anchor cell, and what did I say? Lin3 here is the name of the signal. And there's also a signal which is called the lateral signal between a primary cell telling its neighbors to become secondary, not primary. Okay, so this is a very short introduction so you understand what's going on. Now, how do a biologist learn about the system? So as I told you, they learn by breaking things up and seeing what happens. So for example, one of the experiments goes to this green anchor cell and uses a laser to burn it. And then what happened is that all the cells become a tertiary fate. And from this, the inference that is made is that this signal is responsible and needed in order to push the cells from being tertiary to become primary and secondary. And then you can also refine the experiments and figure out what is the exact timing window in which this process happens. And in terms of genetics, another component in the system is this gene, which is called Lin15. So what Lin15 does, that it normally blocks this a VPC cell from becoming primary. And only if you get a strong enough signal of Lin3, then you'll be able to overcome this inhibition and become primary. So this is an example of, later we'll see in the gene networks, you have activations. So the Lin3 is a kind of activation, and Lin15 blocking a certain fate is called, it's an inhibition. So what happens in, if this Lin15 is not functioning, so you have a mutation in Lin15, what will happen is that spontaneously VPC cell can turn to be a primary cell, and then all the cells basically will race to become primary, and they will also send to their neighbors messages not to become primary but to become secondary, and you'll get these kinds of patterns of interleaving of primary and secondary. So basically, by making these kinds of experiments, this is how we figure out and reproduce a model of the behavior of the system. Okay, so the next stage, I'll tell you a bit about some of the different methods that have been used to model the 3D system. So we'll go quite briefly over that. And I think it's interesting now to see in a retrospective that there's still a lot of work done even to figure out what is the right language, what is most convenient, and eventually maybe there'll be some conversions, but okay, so one formalism, so we're now into computational methods that are used to model the VPC system. So one is this scenario-based language, live sequence charts, which are an extension of a standard sequence diagrams to people that are familiar with. So they can describe kind of rules. So in this scenario, what happens, you say that if you kill the anchor cell, you ablate it like we described previously, then cells will become tertiary fate. So this is like a dynamic rule, and you can describe many different dynamic rules and then run a simulation of the system and observe the behavior. And also you have what is called existential charts. And existential chart, you can think about it like it's equivalent to a CTL, a property in temporal logic that uses an existential quantifier. So it means there is a run of the system that satisfies this kind of behavior. And then the methodology of working would be to describe many of these existential charts, which corresponds to experiments in the lab. And then you would want to check and verify that indeed all these experiments are satisfied by the model, by the rules. And if a certain experiment is not satisfied, then first you try to figure out what is the problem with the model, but then maybe you identify certain things that you haven't thought about it, or certain things that the current theory cannot explain. Okay, so this was a quick example of a model with scenarios. Another approach that was taken is modeling the system using state charts. So state charts are a generalization of classical state machines with orthogonal states that can run in parallel and with hierarchy of states. And then in this VPC model, each of the cells runs an identical description of the state machine, but each one can be in a different active state, and they can communicate. And this was turned out to be quite powerful and useful to model the system. Another approach that has been used is Petri nets. Right, so Petri nets, as most of you know, right, we have the positions and we have transitions, and the tokens that are placed on the position can be moved according to the transition. So it's a well established and clean mathematical formalisms. And then right here you see quite in this picture quite a huge Petri net that also duplicates different. In each cell you have a different Petri net that is identical. And in addition to simulation tool, one of the advantages is that there are mature verification methods, and This allows to reason and verify properties of quite a detailed model of the VPC. And another formalism that I want to tell you about, because this is a formalism that we're going to use in this RAIN method that soon we'll get to, is Boolean networks. So here you see a kind of Boolean network of the different components that are in charge of the VPC system. So as you remember, I told you about 2 components, but in the actual system, there are many more genes that can affect each other and either activate or inhibit, and eventually this will determine this fate decisions, right? So it's possible to build models in many different kind of levels, and this is one of the more advanced. And the Boolean abstraction means that each gene is either on or off, which is of course a simplification, but it's quite powerful and useful. And you can model mutations by fixing a component to be all the time 0 if it's not active, or allowing it to be all the time active if it's what is called an overexpression mutation.
Sorry, can you tell us a little bit about the different types of edges here?
Yes. So I'm not sure in this figure, but in general, there are edges that are activation.
Mm-hmm.
So it could be that here in this diagram, the solid lines are the activations, but later I'll get to that. Activations means if X activates Y, then when X turns on, Y will turn on.
Okay.
Inhibition is that if X turns on, it turns Y off. So
okay.
This will be important soon, so I'll get back to this again. And another thing that you need to know, if there are several activators going into the one gene, you'll need to know information about the logical function, whether it's an or an and we'll get also to that. So not all the models are the standard discrete models, so there are interesting models here that use a ordinary differential equation to actually more quantitatively describe the gradient of this signal and to study it in more quantitative ways. So there are ODE models, and actually ODE models are, I would say, one of the mainstream approach for computational modeling. And another interesting model for the VPCs is a dynamic Bayesian network. It also represents some of the approaches of machine learning. And here you have a kind of initial information about connections between components, and basically by feeding all kinds of examples on the measurements of the system can learn a model, and this is one of the models that will learn. So you have the ability to learn models and then try to validate the model by making predictions about the behavior. So a bit more about it later. So this is, of course, I would say a competing approach to all the formal reasoning, to use machine learning. And also this is quite still quite mainstream and powerful. So we'll say a bit more about this later. So a bit more about the uses. So I told you about some of the computational formalism. So a bit more about the work that was done for VPCs for verification. So in all of the formalism, we still use typically a temporal logic in order to specify properties that we want to verify in the system. In some of the system we also can simulate, but in many we can verify. And so in a paper with Dana Fishman, we also, as part of this research, we categorize all kinds of patterns that reoccur in biological papers. So I'll just show a few of them, then people that are interested can look this up. So one says that if a signal P2 is not present to stimulate a certain pathway, pathway is basically a set of components that are connected together, but the signal P1 is the P3 signal silent. So in one of the papers referenced here, Ecker et al. 2001, this is modeled by saying it's always the case that if always P1 is on and not P2, then this implies that eventually it will not be eventually P3 on. Okay, so of course we know that it's challenging to translate, but this is one of the patterns that is used. A simpler pattern says that necessarily if we start the system from a state Q1, eventually the two signal P1 and P2 will get activated. This is Q1 implies eventually P1 and P2. And a few other patterns. So there's occurrence, of course, exists a path in which eventually P will occur, or something that will not occur, so not EFP. And these properties can also be expressed in LTL, and therefore different papers have used both CTL model checkers and LTL model checkers. This consequence pattern that is used quite often in biology. You can say it's always the case for all paths, always. If P occurs, then eventually Q will happen. There is a way to get to Q. This means that if a state P occurs, then it's possibly followed by state Q, whereas you can also say that this is necessarily followed by using AF. And again, here we have some property that that if you write it in LTL, it has a different meaning. So here you don't have equivalence, and it's also a question, what would be the right biological properties? And this is, I think, reminiscent of discussions in early days in standard verification, what the temporal logic is. And of course, here, part of the goal is to allow experimentalists and biologists to write this down. So this would be even more challenging for them than. Okay, so I think I'll skip some of the patterns of temporal logic. Maybe an important one is invariance, right? For all paths is always P, and a notion of whether a system can get to a stable state. So this would be captured by there exists a path in which eventually, right? It's for all paths is always S. And again, you can express in CTL or LTL. An interesting property that was introduced by Cook and Fisher and colleagues is stabilization. So for stabilization, we ask the existence of values K1 up to Kn such that eventually always each of the observable variables V1 up to Vn will each one have a fixed value. So this means that the system is kind of in a homeostatic state. So eventually it will get into a situation where all the variables hold the same value. So there's been people can look up in this reference algorithms and modular techniques how to compute this efficiently and to figure out whether a given system is stabilizes or not. And often, like the wild type system, the one without any mutation tends to stabilizes, whereas if you perturb the system, if you knock a specific gene out, it brings it out of this stable condition. So this is an interesting phenomenon to study biology. So now we'll say a bit more about some of the other methods and the use of verification for VPC. So life sequence charts have a direct translation to temporal logic. And then actually you can use the verification method to execute the behavior of the system and avoid violation in the system. And the results of a translation to a temporal logic. And here part of the motivation is also to allow a more convenient visual front that it would be easier for experimentalists to use compared to writing the temporal logic.
Hillel, can I ask you a general question? When you're applying temporal logic to a biological system, I'm trying to make the parallel to normal temporal logic and model checking and stuff like this, where you have the non-determinism coming from the inputs. So what are the inputs in a biological system?
Okay, so the inputs would be to some extent the environment in which you put the system in. So it could be in some of the models, the input is if we intervene somehow with the system, if we actively mutate certain genes, or if we put some signals. But in some models, the input could be another part of the system. So in some in models, you can say, for example, in the VPC system, you can look at this anchor cell that sends a signal as an input, and then build a model of the just the 6 cells, and regard this signal from the outside as the input. And then you can basically reproduce experiments in which someone comes in nastily with a laser and burns the cell, and it will change basically the input to the system.
So you refer to this C elegant as something which is completely deterministic. It doesn't have any kind of decision making.
So in that, so I'll quantify it on the high level. That's what I said, but and I said it specifically for the development. Just this process of deciding what type of cell each cell will be. It's almost completely deterministic. There are a few cells in which there's let's say there are a pair of cells that you can either one of them will become one cell and the other one will become the other one, and there's a kind of competition which one will be. So you always have one of the first type and one of the second type, but it's non-deterministic which one it will be. But later on, in terms of the actual when the worm is functioning and doing all kinds of things, there it does have all kinds of stochastic behavior.
I see.
For example, in movements, there in C. elegans or other things, they have actually a very interesting algorithm how they know how to go to the food. So basically, they measure the gradient, how much food there is, and if they see that the gradient is positive, they continue to go straight on. And if not, if they see the gradient is not good, they randomly make a twist and they move a bit more. And then this frequency of when to do the twist basically causes them to navigate to the ideal conditions. This is an example where they use stochastic behavior to navigate.
Okay, I think that Georg had a question. Georg?
Yes, in this context, you've mentioned stochastic behavior. Are probabilistic models used to model these biological systems?
Yes, there's. I'll get to it. Yes, definitely, there are probabilistic models. I'll speak a bit more if I have time in engineered application, but it's very relevant for many of the applications. Definitely. Okay, so in terms of the verification of state chart models, right, there have been indeed usage of either automatic testing driven by verification or actual verification. Part of the challenges are that some of the tools were originally used for embedded software, so they allow to write C++ code and allow to introduce unbounded size variables and create objects dynamically, which makes life harder for the system. So part of there's research that uses reactive modules and the Mocha tool to do verification, and other works in which the language is simplified and you don't allow biologists to do all the things with the C++ code, but you get advantage of being able to verify the model. And for Petronet, as I mentioned, there are many different extension and also powerful verification methods. And to Boolean network that soon we'll discuss in more depth. So Boolean network, as I told you, right, gene would be either on or off. And this is very natural for verification because right, of this logical abstraction. One interesting analysis that is done is to find all the fixed points of the system. So the fixed points are basically these stabilization points. And before I told you that there's interest to see whether the system stabilizes only to one stable situation, but often it has different fixed points, and then you want to compute all of them. And this has been done using symbolic algorithms and BDDs and also computation of attractors. And a bit about these models of VPCs in dynamic Bayesian networks. So, so as I told you, learning approaches are indeed quite dominant in the inference and understanding of gene networks. In terms of advantages, one is exactly related to this point that was asked now, is that they can deal with noise and stochastic behavior. And also the methods are quite scalable. And so in formal verification, we have managed to scale up things, but it's still a challenge to deal with a model that has 50 or 100 genes would be a lot with lots of experiments. And some of the cons of these machine learning approaches is that actually it's harder to identify inconsistencies. That is when there isn't a model that explains the experiment. And from our experience, actually, these inconsistencies have often been the most useful. The fact that you don't find the model that explains the data, it forces you to figure out and maybe identify new interactions or reconsidering assumptions. So it's not always good to automatically get a model. And also another challenge is understanding mechanistically what is the meaning of the model generated. Okay, so I'd like to tell you now a bit more about how we can infer gene networks. And let's look at a simple model, which will be a Boolean model. So over here, we have a small circuit. And here, there are two types of arrows. All of them are going to be activation, but the dashed one means that there's an interaction that we're unsure whether it exists or not. So there's some experimental evidence that there's this interaction number one between KLF4 to OCT4, but it's not completely sure, right? And in biology, it's many times the case that there's evidence, but it's not easy to reproduce, or it depends on the situation. So, or think or not white and black. It's not a mathematical proof. Sometimes you have all kinds of hints or suspicions. So it's typical to have a situation where you have these, so here we have four interactions that we suspect might occur, but we don't know. And we would like to infer a network, to infer a model. And how can we infer it? We infer it based on experimental results. So here we have two different experiments. In experiment number one, eventually the system gets to situations where all the components are on. In experiment two, you have a different pattern. Okay, and here the green nodes are kind of signals to the system, but it's right. This is not an essential. Okay, so basically we have here hidden 16 different possible models, right? You can each interaction either include it or not. Right, so you have 16 different topologies, and you can ask which of them will satisfy the different observations. Right, so here are all the different possible models, and for this example, it turns out that only 6 specific models will actually lead to these experiments. So this is for a simple model. Of course, this is an example of an initial model of an embryonic stem cell. So embryonic stem cell, it's a bit like the VPC system that we discussed. It's a cell that is still not differentiated and will later turn into a blood cell or heart cell, and this is a part of the core circuit. So we have a lot of interactions that are not completely we're sure about. And of course, we have a huge number of models. Right, so it's this of course is a challenge, but of course with formal verification methods, for us, it's not such a big deal. We can deal with this kind of combinatorics, and we can figure out in this way. So it was colleagues at MSR Cambridge have been able to figure out this core circuit of mouse embryonic stem cells, which basically decides when this cell will remain an embryonic stem cell and when it will begin on its path for differentiation. So very on the high level, what is done is you start with this network that has many undetermined interactions. It also has some, you see some lines that are not dashed, that are full, and they are the ones that we're more confident about. And we have quite a large set of experiments. So in the bottom of the figure, each basically column over here represents an experimental measurement that was done in the lab and measured how much each gene is activated at steady state. This allows to reduce and to find different solutions that are consistent with all of these experiments. Right, so this synthesis approach, we're able to basically encode the problem into an SMT problem and look for only the networks that satisfy all experiments. So what kind of things can we do with this ensemble of networks? Typically, there are more than one. If it would have been only one network, then we can just run a simulation and check. But if we have many networks, one question that we're interested to figure out is to know what will happen if we perform mutation in these two cells that are denoted in X. So we want to figure out how it will affect two other genes, OCT4 and SOX2, that are positioned in the middle. And these two genes are known to be the main factors needed for a cell to remain an embryonic stem cell. So basically what we can do is we can ask whether all of the models that we synthesized, whether all of them agree about the value of OCT4 and SOX2. And if all of them agree that they'll be on, then we make a prediction that they'll be on. So this allows a systematic way in many of the cases to make new predictions about the dynamics of the system. And this turned out to be very powerful because many of these predictions were indeed turned out to be correct experiments. So I'll try to summarize a bit it's the proper formulation for the synthesis. So we have a G is the finite set of genes. E are the solid lines. These are interactions that we're positive about. E with a question mark are the optional interactions. And both of them can be positive or negative, means either it activates or it inhibits. And R is a set of regulation conditions. So regulation condition basically is a template that determines the logical function. So this is a table that explains what are the regulation conditions. So basically we have a family of, here there are 18 different functions. Let's maybe look at the line number 0. This is one of the regulation conditions. It says that the gene in the middle will turn on if both of its activators are on. So the red color means it's on. So in this regulation condition, the only way for me to turn on if it's all my activators are on and none of my inhibitors are on. And in function regulation condition number 1, I can also turn on if some of my activators are on. And we have in this way, we take all the functions that are monotonic. That means that if I have more activators, I'll remain active. And if I have more inhibitors and I'm off, I'll remain. So this is both makes sense biologically, but also it turns out the synthesis problem to be more feasible for the SMT solvers. And the synthesis means I want to select optional interactions and select a regulation condition for each of the genes in a way that all the different experiments are satisfied. That means I'll have one run for each experiment. This basically means that we found a network that can explain all the data. And as I told you before, then we can do predictions with the system. And we've developed the RAIN tool that allows to basically write a kind of programming language representation, write these networks and the constraints, and it uses under the hood a translation to Z3 and a bounded model checking problem to find out these interactions. And it's quite it's still of course a challenge to deal with many components, but we can handle 20 or 30 components and hundreds of experiments. So this turned out to be, this is an online tool that we've been using to study several systems that are shown here. And I won't get into more detail about the system, but part of the interesting thing that you can do is also identify new interactions. When you get a situation where the solver says there's no solution to there's no network that explains the problem, you can add additional optional interaction and then ask whether it can now find a network. And you can ask, for example, for the minimal number of interactions that needs to be added. And this also turned out to be a useful approach. So it's an approach how to identify interaction between components, which is something that biologists are very interested to figure out. Because any new interaction, it's a new way to understand the effects in the system. In some systems which have more medical applications, new interaction can also be utilized as drug targets. So right, there are groups and researchers that are now trying to do more applied research, and they use reasoning methods in order to identify a drug target. Okay, so so we have these kinds of complicated networks. And one of the, I think, exciting discoveries that was made by Uri Alon's group at Weizmann is this notion of a network motifs. So what is a network motif? It's a small graph within these bigger networks that appears many times. So they did statistical analysis and found these triangles or all kinds of circuits that you see over here. This is called, for example, a feed-forward loop that is enriched in the graph. That means if you would compare it to a random graph, it would appear the several standard deviations more than a random graph, and therefore probably it has some function in biology. And by analyzing it using more differential equation methods, they were able to figure out roles for some of these systems. And it's also kind of a handle to try to break the complexity of the networks into more manageable parts that we can understand. So one extension that we've been doing for the RAIN tool is to allow to specify in these networks that are still partially understood with many optional interactions, to specify these patterns of motifs, and then ask ourselves whether there are certain motifs that must occur in the system. So these are some of the results, and then you can extend it. You can ask, for example, about combinations of motifs, or you can ask whether a certain motif must not occur. And we're trying now to apply it to additional biological applications to really try to understand what are the building blocks of these networks. Okay, so in terms of future research, of course, there are many challenges in terms of handling a bigger networks. So these are gene networks that are of 50 or 100 genes, and we're facing many difficulties to handle these completely automatically in synthesis. And another interesting direction is that during development, over longer periods, cells actually reconfigure their network. So over here, if you look at a path of a cell, when it was here, when it moves here, the network changes a bit. So some interactions function and some interactions stop to work. And this is what is called the model of a switching network. And I think it's interesting also to compare to standard computing methods. Initially, when in the first architecture, it was possible to write programs that are self-modifying. And actually, it was quite useful because there wasn't enough memory, but now it's quite esoteric. No one wants to change. We hardly can debug our problems without changing them. But biology seems to use this trick of modifying the program while it's run. Okay, so I think we're running a bit out of time, so I want to get to tell you a bit about the part of the engineered biological system. Right, so why would we want to actually build the new computational devices from biological material? One of the dreams initially was that maybe they will be really fast and can beat standard computers, and they're still interesting in this field, although it's quite clear that this is very, very hard. So it might happen, but it's not going to happen in the next few years because the technology is very mature and it's a very challenging problem, but there's still a valid research directions. And another motivation is energy efficiency. So maybe we can have machines that are slower, but they're more efficient, and this can have all kinds of applications. Another motivation is that we build these devices in order to understand biology better. Because once we start to build things, immediately they don't function as we want. Things are messed up, and then we need to figure out and sharpen our understanding of the biology in order to build the circuits. And in terms of applications, I believe that actually the main applications might be in diagnostic and medicine, and not necessarily replacing standard computing. So there are many, many different approaches to do these kinds of artificial biological systems. I'll tell you a bit about strand spacing systems, which use basically the DNA strands in order to program material and build small programs. So what you see in this slide, you have strands of DNA, and strands can stick together using this complementarity. So when A can stick to a T and a C to a G, and over here, these are short-stranded, so they can short strands, so they can bind and they can also unbind. Whereas if the strand is longer, then it's just eventually attached, and then it will stay together for a very long time. So this is how you can design an AND gate using strand displacement systems. So we have on the bottom, we have actually the gate, and we have the two inputs. So as you remember, strands can bind if they have complementary sequences. So here, the first input is bind along this red toehold. And now it's in the right position to replace the upper strand. And then it kind of does a random walk. And you see that the strand has gone into place and displaced in the middle strands. And now stochastically input 2 can also come into the system, connect to the other toehold, and then eventually again through a migration, it will catch the place and will completely displace the output. And the output, once we measure the output, we know this is basically the result of the computation. This means that we have both input 1 and input 2, and sometimes we can also measure it. So the way how we describe this mathematically is using a chemical reaction network. So on the left, you see a CRN equation. So you have here X is one of the inputs. If it comes together with G, it can form this complex XG. So it's connected to the gate, and the same for Y. And then if XG binds with Y, then you get that X and Y are connected to G, and you get the output. So these are two ways to get the outputs, and this is how we can represent the system using chemical reactions. And chemical reactions are basically used for a powerful both modeling language and simulation language. So I'll show you also a quick video that shows what's going on in one of these strand displacement systems. Okay, so this is how things happen in actual 3 dimensions. So chemical reactions, as I told you, can be used as a kind of a programming model. So let's see a few simple examples just to clarify. So suppose we want to compute that Y is 2 times X, and we want to write a chemical reaction equation in order to program this. So the program would be that in X goes to Y plus Y. So in X goes to 2 Ys, and here we double, right? We double the number of X's, and we get eventually, after all the reactions are fired, we get the required result. Another specification might be we want to divide X by 2 and take the floor operator. So here we'll just make 2X bind together and create a Y, and this will exactly compute eventually all the X get annihilated, create the Y, and you get half the number of X's. You can also do the sum of X1 plus X2 easily by having 2 equations Each one will take x1 will turn to y, and x2 will turn to y. And you can also take the minimum to compute the minimum of x1, x2. Right, so basically x1 and x2 go to y, and then this process will stop when we have used the minimal number of x1 and x2. There will be no more possible reactions, and the y will be exactly this number. And you can also do more complicated programs like the maximum. So the maximum is basically computed by doing the sum and subtracting the minimum. And this can be done by these four reactions, in which first x1 is copied to L1 and creates a y, and the same for x2. And then you do a minimum by doing L1 plus L2, which goes to k. And then you subtract this minimum from y. So y plus k gets annihilated and vanishes, so it gives 0 molecules. And in this way, you will compute the maximum. Okay, so you can do all kinds of tricks with chemical reaction network. And so this is one interesting program. So we can ask ourselves, what does this thing compute? So we have x plus y goes to x and this new b. Or alternatively, if you have y plus x, which is the same thing, you can have again a y and a b. And then each time an x meets a b, it will turn to two x's. And when y meets a b, it will turn to two y's. And in terms of what happens actually in this computation, think about it as kinds of molecules right, bumping into each other. So the rate of this behavior will depend on how many x's and y's and b's you will have. So the rate for a certain reaction to occur is dependent on the product of its the number of molecules of each one. So what it will do, it's actually the system can compute the approximate majority. Let's see a quick illustration of that.
Expresses three reactions. First, the two populations x and y can cancel each other out. Then whichever population.
Okay, so this was the approximate majority, and a visual DSD tool developed at MSR Cambridge by Phillips and Luca Cardelli and colleagues allows us to program these kinds of systems either using strand displacement or directly in chemical reaction and convert them and run a. So here you see a simulation where we start with 30 x's. So the x's are the red ones and green are the y's, and you see initially there are all kinds of b's created, but the x dominate and you'll get to, I think this is the stochastic simulation of the system. Here on the second slide, you see another simulation, and again you get in this case the majority of x's, but it's the time dependence and the order in which this occurs is different. And right, if you'd run many simulations, if there's the gap between the initial x's and y's is not so big, there's a chance that actually the y's will dominate. And there's actually an analysis that shows what is the probability for this to happen. And these are actually population protocols that were developed and discovered by Anglo and colleagues in an independent way. So another kind of semantics for these systems is when you have a very high number of molecules, the useful semantic would be a continuous semantics, which use an ordered differential equation model, and you can, within the tool, also run these kinds of simulations. And of course, as we now, you already see that even with simple models, it's quite tricky. So when we program these models of strand displacement systems, it's really essential to be able to not do only simulations, but do formal verification. And indeed, here it's probabilistic issues become important. The One of the approaches uses a PRISM, which is a probabilistic model checker, to encode the system and to be able to verify correctness properties. Here it's kind of a CTL properties that ask whether in this circuit, whether you get the correct output. And you can also ask, in some of the properties, although the system is probabilistic, but you have eventually you'll get always to the right answer. But in some cases, either there are errors or inherently the protocol is probabilistic, and then PRISM can give you an analysis of what is the probability for something to happen. So here we see some results in which you get a graph that shows after a certain time whether the circuit is terminated, and if it has, whether it's in a success or an error state. So this is very useful, but indeed there are many challenges. Once you build the more complicated systems, it's very hard for the verification tools to cope with it. So there's exciting future research to do in the field. One of the analyses that we did on a non-probabilistic property is this square root circuit, which is one of the big systems that was built using strand displacement system. So here, what you see in the figure, you can see the details. These are many of the all or part of the reactions in the system, and we translated them automatically to basically an SMT encoding, and then we're able to use a Z-tree and be able to prove correctness properties of the circuit. So people that are interested can look up more details about this work. Maybe one interesting thing that as part of the proof, we had to use inductive invariance, where the invariants are a property of conservation of the strands. So for example, in this reaction, when strands come together or break apart, you still have the same number of each of the strands. So you see you have here one strand that is a B and a TX, and here it's on the other side. So things can stick together or fall apart, but you don't change the quantity. And these kinds of invariants were useful for allowing the proofs to go through. And potentially, this is a conjecture that maybe also in real biological programs, what allows them to be robust is that they have these kinds of invariants in the actual program, in the actual strand. Okay, so we're a bit short of time, so I'll just say very briefly about another approach that allows to do a engineered biological system. And here, just in the high level, what we do is we manufacture these kinds of networks using standard lithography techniques. And we put biological motors that can just drive through this network. And according to the place where they arrive, we can compute the answer to some problem. So for example, consider the subset sum problem, that you have a set of numbers, in this example, 2, 5, 9, and you ask whether 11 is a possible subset, and it is, because you can select 2 and 9. And 13 is not. So this is what the subset sum problem is. How do we encode it in these systems? We have this network in which agents come in through here, and they can go either down or diagonally. So let's follow this yellow or orange system here. The system turned diagonally, so it counts 1, 2, and now here it has another split. It goes down, it means it's not going to add 5. Now it gets to another split, which means it's going to add 9. So it goes diagonally and it goes out of exit 11. So it means that 11 is a possible subset of the system. So the whole idea is to put in to miniature this whole thing and put in many biological agents and allow them to traverse these kinds of mazes and networks, and to then measure the exits, and according to that, we can get the results of the computation. So this is really a pure mathematical computation. And I think I'll skip some of the experimental details. We have encoding for other kinds of problems and other junctions. and all kinds of optimization to make things work more nicely. And then we've developed a formal verification tool that allows to basically encode these networks. And once we have many optimization, to still be able to prove correctness of the design. This is really important because now when we colleagues and collaborative groups manufacture the system, we have many physical errors and measurement errors, and we want to at least know that logically the system is correct. So these are some results of correctness of these circuits. In terms of outlook for the future, I think one of the exciting things is that for gene and network, there's I think a good chance that the formal reasoning methods will become a much more mainstream and important and used by biologists. And of course, it depends on whether we can further scale the methods and make them more usable. One challenge in terms of the research is building what we call whole tissue models. So in the gene networks, as I showed you how we synthesize, we focus on one cell. But then we would like to prove properties of many cells that function together, and this will probably require new compositional methods or ways to deduce from the behavior of one network how it affects other networks. And in terms of the engineering applications, once there are more industrial applications. So currently still a lot of these ideas are research prototypes, but potentially when some of these applications become industrial and more mainstream, there will be a big use for being really confident and being able to verify the correctness of the system. So I think this is what I wanted to cover, and thanks to many collaborators, and thanks for you for listening.
Okay, so thank you, Hillel. So let's see if we have questions. Yes, Muhammad.
Hello, can you hear me?
Yes, yes, yes.
Okay, perfect. Yeah. So I have actually a very general question. So for the type of DNA analysis that you showed, I assume that privacy and security will be of great concern, as in the medical data remains protected, right? So I was wondering if you can perhaps comment on some formal verification efforts for security and privacy of such DNA analysis, please.
I know it's not really directly connected to things that I showed today, but I know that there are many applications in which you, right, there are frameworks that allow you to, for example, if you want to do the analysis on a specific data, you can anonymize things or run homomorphic protocols that you don't know on what is the actual data that you're doing the measurement. So I know there are several research efforts and even prototypes that allow to do these kinds of things, but I haven't personally worked on this. But it is indeed a big issue in order to, for example, for hospitals to share some of these data, right? You need ways to how to compute these things without showing. But I haven't worked, so I don't have specific answers for this.
Okay, sure. Thank you very much.
Yeah, I see, Warren, that you have a question. Do you want to raise the question?
Yeah, hi. Thank you for your talk. Interesting, challenging. So I'm just curious if you could put the FMCAD community to work to help you, what new techniques or methods or theories or whatever would you like to see us work on to extend your capabilities or improve your precision or quality or reach or whatever?
Okay, so this is a great question. I think one of the questions is indeed, for example, for this synthesis question that I showed, scaling it up to 100 or hundreds of genes would be very useful. And actually, I was thinking in the past, maybe one way to encourage research in this, maybe it's possible to arrange some competitions where we provide benchmarks of existing networks and simplified format and organize something along. So that's one direction. Another one would be this compositional techniques for if you have many different networks for several cells, how can you combine them to reason about the system? And the third one would be the probabilistic analysis, which is still very challenging.
Mm-hmm.
But indeed, I think one of the challenges is how to phrase a question that would be easy for easier for the community to access, and maybe it's through providing a challenge benchmarks where we kind of annotate and say what is the property and what we want to figure out. And I think we have by now many problems that are even beyond the scope that we can tackle, and maybe this is a possible way forward.
Mm-hmm.
So I wonder also if you have the problem of, you have a hammer, so things look like a nail. So you have a lot of, I'll say, state-based exploration techniques. But I didn't see, I would say, more general programs that might be used. For instance, recursion or, some more richer machines where you might make greater use of inductive invariance or to decide some properties possibly by induction. And is that because it's richer than the community wants, or it's just too hard and the models are too big and so it's not something that your community investigates?
I think there's still many things opportunities that are unexplored. So there could be that there's some work on this already, but I think that the field is still, as I try to convey, there's still a challenge in terms of building the models and figuring them out. So I think these are definitely worthwhile things to check out.
Okay, we're actually out of time for this session. So perhaps now, thank you very much, Hillel. And we'll go for a coffee break for 10 minutes, and then we'll continue with the last session for today, a tutorial by Armin Bire. So see you in 10 minutes.